Thank you to Dr. Renat Henderite and Professor Hadi Suwono. Now we arrive to our final session, and I would like to call for the fifth moderator, which is Dr. Reiny Antoneta Tumbol, which now is a lecturer at Faculty of Fisheries and Marine Science, Ratulangi University. She had her doctorate in the University of Queensland, Australia, at the Center for Marine Studies, Faculty of Biological Science.
Okay, distinguished participants of Icon Smart, ladies and gentlemen, we're going to move on to the next plenary session, and I'm Reiny, and I'll be your moderator for this session. And for this session, we're going to have the last speaker for plenary session. According to a theory that save the best for last. So I hope this is the last speaker, and we're all going to be excited to get this information from our speaker. And I would like to invite Professor Dr. Zulqarnain Mohamed from Universiti Malaya. Welcome, Prof. Before we start with this session, I would like to read a little bit of the Prof. Zulqarnain CV. Prof. Zulqarnain bin Mohamed, professor and a dean of Institute of Biological Sciences, Universiti Malaya. And his expertise is in human genetics, genomics, molecular genetics. His academic qualification, PhD in human molecular genetics, University of Aberdeen, Scotland in 1999, and Bachelor of Science and Honors Genetics, Universiti Malaya in 1994. And he has published numerous of articles in some recognizable journals. And today, Prof. Zulqarnain will talk about marine mammal conservation, DNA-based approaches to studying biodiversity of selected dolphin species in Malaya. Time is yours.
Right. Thank you very much, Dr. Reiny. It's of course very challenging to talk before lunch, and I've also been given about just 15 minutes. Mohon izin saya dari sini ya. Okay, thank you very much, Icon Smart, for inviting me. Thank you, MIPAnet. Thank you, MDSM, for the support. Right. Just a little bit of disclaimer. Yes, you did hear it right. I am a human geneticist, but why I'm talking about dolphins has got to do with my schoolmate. I don't think it's reaching the. Can I have this? Yeah, it's not reaching the. No, next slide, the first slide, okay. Right, it all started with this guy, the guy in yellow, so he's my schoolmate. So he was talking about his work, he's very experienced in marine mammals. And it was just one line of a conversation that says, are you not interested in genetics? The next thing I know, I was in a boat and then out to sea and then looking at the marine mammals. Next slide, it's not working. All right, so we always hear about this in the news, about extinction, about the recent one, I think this is 2009, a Sumatran rhinoceros, the one that is in Malaysia died. So it is very profound. And next slide. But I can assure you extinction is also a normal biological process. Extinction should be balanced by the emergence of new species, and that's how life goes, right? Unfortunately, this is the recorded extinction. Next slide. Now, unfortunately, this is the concern of biologists, is that the fact that the rate of depletion is at the moment far exceeds the emerging of new species. And unfortunately also it is a lot to do with human development, right? Human activities. And then it is alarming that the current rate is about 4 to 5 times higher. Next slide. So in talking about conservation, there are two major aspects that we want to see. Is that one of the first ones is that we need to determine exactly what is the threat, what is the source of the threat. And secondly, we need to know what we're dealing with. We need to know exact nature and what species and so on. We need to understand the species so we'll be able to help conserve it. Next. So in genetic diversity, what we can do using genetic analysis is that we can actually determine the history. If we're able to get the samples, we will be able to determine the history and then look at diversity. And why is diversity important? Because loss of genetic diversity reduces the ability to adapt. One simple example is the COVID-19, the pandemic. You see that some of us got infected, but they are mildly affected. But some of them, they have very severe symptoms. Now, this is about the genetic background. And similarly, when we receive vaccination, some of us react quite adversely, but others don't feel anything. So we need to have that diversity in the population, in the human population, and also in our animal population. Next slide. As I mentioned, human activity is contributing towards the loss of diversity, and sometimes it's not really something you plan to do. It's because of human development, the needs for recreational, the recreation, the needs for economic, the need for habitat and so on. Somehow it drives the reduction of that diversity. There are, of course, other factors, for example, environmental changes, the climate, but unfortunately, again, the human effect is also affecting the stochastic factors, the environment factors, so we are contributing doubly into the loss of genetic diversity. Next slide. So again, what do you do in molecular genetic analysis? Again, you get the samples and then you study the social structure. You study the family diversity, and from the diversity you can the Brunei Bay, it's between Sabah, Sarawak, and also Labuan, that area. And then the other one is in Perak, Kuala Sepetang. Next slide. So this is me and my friend. So this is our preparation of the darts. Next, the one before, please. No, that's going too fast already. One before that, all right. And before you get any ideas about, how nice it is to work in the sea, and if you watch a lot of National Geography, It is far from what we have. We have that very small boat, and there are about 6 of us, and then we sit in the hot sun without any shelter, and then we just look out and then for the dolphins, right? So that's our boat. Next one. If we're lucky, we may have some netting above our heads, and then if we're also lucky, we may get some boats that that has a roof of that sort, But other than that, mostly it was just us and the clouds. Next. Now, this is what we do. That's me shooting, but I can assure you we don't kill any of the animals. No, this is for conservation, right? But we need to shoot the darts. If you can see the darts, after we shoot, the dart will fall off. It will actually, there's a floater, fall into the sea. So we retrieve the dart, and then we open up the cutting head, and then you'll get that a piece of flesh and muscle, and that's where we get our DNA from. And then we make a practice that after we shoot, then we'll stay for a while to make sure that the animal is still, well, at least alive, because that's the whole idea of conservation. So this is textbook. We don't just shoot anywhere. We don't shoot the head and so on, but this is like you can see the dorsal fin, and then you can see the small red thing, so that's the dart. That's where you should hit, but I'm telling you, it's a moving target all the time, and then you only see like 1 or 2 seconds, and then you just have to point and shoot. Right, so this is the textbook case. So that's just me. Again, you can see, because the time when the dolphin appears and then submerges again, I looked at the video, it's less than 2 seconds. And then I'm sure, you know how brilliant dolphins are. I'm sure that they're always pulling my leg, because you look at the right, they will appear in the left, and then when you look at the left, then you appear at the right. This kind of work appears a lot of patience, muscles, because the gun is heavy, you can't point all the time. Patience again, heat tolerance, because you're doing it under hot sun. You have to have reflex, because you wait, and then when you see something, and then you just try and shoot. Patience again, accuracy, patience, focus, because the boats, sometimes have a lot of people at the back, and then you're focusing on one place, and then they'll say, left, 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 and then you look and then the dolphin appears on your right. So it's not easy. Now, this is what we've done so far. So we've started 2018. We've got about 20, there are 2 main species, Ocular brevirostris, and then Susachinensis. Funnily, on the Bay of Brunei side, we find a lot of Irrawaddy dolphins. But on the Kuala Sepetang side, we only get a lot of Susachinensis. They are both there. I've seen Susachinensis in Brunei Bay as well, and I've seen Irrawaddy dolphins in the Kuala Sepetang, but their behaviors are so opposite. The reason why we can shoot these animals is because some of them are friendlier towards the boats. So in 2 places, these dolphins behave opposite. So number of samples obtained so far, so I just calculated it, so that means approximately we've spent about over 900 hours worth of surveying and looking for this, and then we got about 37 samples, so one sample is about 24, more than one day. So this is an DNA extraction. We do sexing. This is something that the visual surveys cannot do on the boat. You just look on the binoculars, you won't be able to see it, but with DNA you can tell. And from our data, it's quite worrying, because most of the samples that we got, in fact, all of the samples that we got were females. So we're not really sure why. We thought our methods were wrong. We took the DNA and then we sequenced. We confirmed the results. Yes, most of the samples that we got were females. Next slide. This is just some technical slides. So when we get the sequence, we put them together and see whether we can distinguish between the different fishes, different dolphins and different species. Next slide. Next slide. Next slide. All right, so this is what we got so far. So these are the 2 main species. You've got the Irrawaddy dolphins at the top, and then the Susachinensis is pinkish in color. And then we found that there are 5 genetic types, which suggests that there are 5 families of Irrawaddy dolphins. And then you can see that when we downloaded sequences, I think from India and, from various, and from Thailand also, from Indonesia. So it seems to show that there is some correlation, right? That this is still, study is still ongoing. Next study, and next slide. Unfortunately, with the Susa chinensis, the Indo-Pacific humpback, the results at the moment is it's not very convincing. I'm not sure if I have the slide here. Go next slide, please. So this is a paper from Australia which already suggests that for Susa chinensis, for the Indo-Pacific humpback, there is a lot of variability. There are still questions on the species relationship, so that explains why our DNA sequence is not easy to match with whatever is available, right? Okay, next slide. Now you look at the numbers up there, we'll see that our average heterozygosity is at 0.4 and average number of alleles is 2. This is very, very low and signals a red light to our population, right? But I cannot conclude anything just yet because the number is still quite low. We hope to get more numbers, and then we hope to be able to establish this, and then we can work with the fisheries department and tell them what we can do next. Next slide. Now, okay, you can't see much from this one, but I think you can see this is our more recent work. Next slide. Now you can see here, all right, at the top, yellow and green, these are the Irrawaddy dolphins. And the yellow bits are the ones that we found in Malaysia. And we found that they are quite close to the ones in Vietnam and in Thailand. The green is actually from Indonesia, but it's more on the Kalimantan side. These are sequences downloaded from the database. Grampus is the second at the bottom. Is actually called Risso's dolphin. It looks quite similar, but it has a larger dorsal fin, and I think there are some cases where the published work shows that it's mistaken identity. So you can see the clustering, right? The sequences will allow you to cluster different species together, and that confirms the identity. And also, because mind you, when you're at the sea, you only see this for a very short time, right? You don't get to catch it and so on, so it's not really easy to determine the species. So DNA collection tells them, confirms it. And then our DNA evidence also contributes to determining the identity of stranding animals. Stranding animals are kedamparan. These animals, they are found on the beaches and so on. Sometimes the morphology is so deformed, so you can't tell which is which. In fact, as a matter of fact, one came in as Irrawaddy dolphins, but when we did the analysis, it's actually Risso's dolphin. And so the other one was finless poppers. These are, they look very much alike, but DNA analysis will be able to tell. When we told them that this is Risso's dolphin, they were very excited because they've never seen visually a Risso dolphin before, right, around the area. So DNA evidence actually allows you to do this kind of thing. Next slide. Now, this is more recently. It'll probably just take another 2 or 3 minutes. What we do now is we look at environmental DNA. Why do you want to do this? Because we want to see, other than looking at the dolphins, we only want to look at the environment. So how do we do this? Next slide. So here you can see this paper that says harnessing the power of eDNA metabarcoding for detection of deep sea fishes. Basically, what it's trying to say, this is a paper from, I can't remember which group. Basically, what you can do is just you take like, in the paper, you say like 1 cup of water, get all the DNA out, and then you do metabarcoding, and then you can tell what's inside. Next slide. So basically, because in the water you'll have fragments, DNA fragments come from the skin cells, from the excrements, from tissues and so on. You can get that from eDNA from the water, and then you can filter it out, and then you can do the biodiversity of that particular area. Next slide. This is the traditional way is on the left, where you have to do a bycatch analysis, what's available. But now we just take water, we filter it, and then we sequence it. This work started quite recently, about 2 years ago, and I think it's giving us quite interesting results. Next slide. So this is how we do it. get water, filter, sequence, and then analyze. Next slide. So this is a Niskin bottle. This is a very heavy bottle that you lower down into the water to get water you want to be consistent. So we took water, it's called surface water, what about from 1 meter. It's a very heavy bottle, but my colleague, the Saiful is in Marine UMT. They have it, we don't have it, so we had to borrow. And they were making a lot of noise because it's very heavy. Next slide. So, part of being innovative is trying to design this. Well, I can't say it's my idea, it's already on the YouTube. We can also use the pipe, the sewer pipe, and also the plunger for the toilets. And then you have a pin, so you can, and then you use a bungee cord, right? And then you can pull the pin and then it closes. It works, all right? So it works fine. In fact, the other day we went out. They were using this more than the official Niskin bottle, because Niskin bottle is so heavy. And because it's so cheap, we also call it Miskin bottle, because we don't have money to buy one. So on the right is the cup that contains a filter. At the blue bit, there's a piece of filter, so you put water in, and then you have to pump it out. This is one of the first times we did it. We used a manual pump, right? But now we've gone some advanced. We use electrical pump, and there's some advances to it. So we don't. This is actually done on the boat, but now we actually bring it back because we have means to do it now. Next slide. So this is one of our tests. We did sampling in 3 areas. We took 2 samples from there is sighting. That means when we actually saw dolphins. And then 1 sample from where we know doesn't have dolphins is in the river. Next. Next slide. And then this is interesting. So this is the array of different animals that that we can find in DNA. So this is very, very new data. So we have yet to confirm it with the fisheries, with people out there, whether this is correct. And then we are comparing. But we are able to compare between these 3 sites, A, B, C, which one contains what, do they mix, Why are we interested in the fish biodiversity? Because we believe dolphins are there because they want to eat. So perhaps the different diversity have an influence. And because there's also a lot of fishing activities, the fishermen and so on. They are supposed to do their work like 5 miles offshore, but unfortunately, they do this very close to shore. So we want to see the diversity. We suspect that the diversity has reduced greatly in over the 10 years, because they've also reported that the amount of dolphins have also dropped. So we're trying to correlate with the fish diversity, and study is ongoing. Next slide. So we can compare between the A and B and see what is unique in A, what is unique in B, what is shared in all 3 localities, and so on, right? Next slide. We also found human DNA as well. Well, of course, there are a lot of fishermen doing it within the area, so they do a lot of business there as well. We've seen ducks, we see mollusks. Next slide. We do 2 types of analysis, because the power of this work is also dependent on how you analyze your samples. So we have one established by the company, but we also do our own analysis, and then we get slightly different results. And in terms of what the diversity is. Next slide. This is the mollusks and the anchovies that we find. A lot of anchovies there. Next slide. And we also find, this one I can't really explain. How do we find a cow's DNA in the water? Maybe we went for a swim. But I can explain why we get macaques, because this is from the river samples, and then indeed there are a lot of macaques even swimming across, so we did get their DNA as well. So it's actually an exciting new thing that you don't have to sit there, you just take water, filter out, and then you know what's biodiversity. Next slide. So challenges, we need to yet determine what's the best volume sample, what's the best sequencing sample, what markers to use, because it's very, this is very molecular genetic space. You have to do a lot of tests, but I think we're getting there at some point. Yeah. Next slide. These are the people who's working with us. On the right side, that's my colleague with his graduate master student, and also the master's student on the red cap. In the middle, Dr. Song and Dr. Sally. Dr. Sally used to be my postdoc. Dr. Song is working with me now. The other two on the left, they are students. That's why they don't look very happy, because they have the slides, they have their thesis to write. And I think this is where I end. I think next slide. Is there any other slides? So I acknowledge the Ministry of Higher Education, TUMEC, University of Malaya, University of Malaysia Terengganu, and also First Institute of Oceanography from China for giving us the grants, Department of Fisheries as well for allowing us to do this work, but most of all, IconSmart for allowing me to give this talk. I leave you with this, my number and my email, so some pictures that we took on our excursion. Thank you very much.
Thank you very much, Prof. And very interesting talk indeed. I'm sure we are all looking forward for having lunch, but that's why I'm going to give only 1 or 2 questions. If anyone would like to ask questions, otherwise I'm going to ask 1 question, because you mentioned that most of the samples, the dolphins, were female, okay?
Yes.
Recording stopped.
Would it be related to the fact that maybe male are more difficult to get close to human, or the female are in their stage of pregnant or something, and they're so hungry and looking for the food around the boats or something?
I think you've given all the answers. Well, to be honest, I'm not much of a behavioral animal behavior research, but I've spoken to some people. Males are more wary of people that or disturbance in the area, so they are very suspicious. And females tend to be more friendly, and, you know. So and then they also bring their calf around just to swim and so on. So they tend to be more open to people encroaching. So that's what we think.
So could it be confounding factors then for your research? What do you think?
At the moment, it's really money. And also, I wish that I can develop a way to call them and then stay, and I can just get some samples, because it's been very, very difficult to get samples. Because you really wait. Because as you can see in the slides, there are some days that we just went there and we didn't see any. There's no way as such at the moment to predict the presence. You're talking about this whole wide sea, and then you're just guessing. So that's been a challenge.
Okay, thank you very much. Let us give a round
Thank
of
you.
applause to Prof. Zulqarnain.
Recording stopped.
And I'm going to give this back to MC. Thank you. Thank you, Prof.
Thank you to Mrs. Dr. Reiny and Professor Zulqarnain Mohamed. So this concludes the end of our plenary session. We now enter to lunch.